seventh lesson. Okay, children. The world of microbes. What is the name of the lesson? That is the world of microbes. Okay. In the previous class, we have talked about the microbes. How many types of microbes are there? Unicellular, multicellular. Yes. And cell clusters are there. And what is a cell? Where do you find a cell? And in animals and plants and human beings, and there are so many cells in our body. Our body is made up of number of cells, and we also studied about the who invented the cell. Okay, who invented the cell, and also who made a deep study of the cell. Okay, and we have seen that some microbes are useful to us and some microbes are harmful to us. Okay, children. Yes, we studied in the previous class. Now let us go to the next part of the lesson, that is where do microbes live. Okay, where do the microbes live? Yes, microbes. Where they live. Microorganisms are widely distributed in nature and exist as unicellular and multicellular and cell clusters. Some microorganisms are beneficial and some are harmful. Okay, the major type of microbes are bacteria, fungi, protozoa, algae and virus. So here how many names they have given for the microbes? They have given 5 names. We have 5 types of microbes in our body or in our nature or in the nature. There are how many types of microbes? That is bacteria, fungi, protozoa, algae and virus. These are the 5 types of microorganisms. These are widespread in the nature. Okay, they exist in the nature, and they are some are unicellular, means only one cell, and some are multicellular, and some have so many cells clusters in the body. Yes. In the next part, let us see bacteria. What is bacteria? Bacteria is a unicellular organism. Bacteria, it is a unicellular, means it is not a one cell, it is having so many cells. Unicellular organism. The cells are described as a poriotic because they lack of definite nucleus. They do not have a definite nucleus, so they are called as a poriotic cell. Yes, activity number 7.2. In the activity, he has told about the bacteria. Let us see here. Take a small amount of curd. Yes. Hal hakidaga there are some bacteria which change the milk into curd. That bacteria, it is useful to our body. Yes, like that. Some bacteria are useful to our body and some are not useful to our body. So take a small amount of curd, put a drop of curd on a clean micro slide. Okay, it's telling you to take a little curd, put on that slide. In the glass, blood test you blood glass ಮೇಲೆ blood ಹಾಕಿ ಚೆಕ್ ಮಾಡ್ತಾರಲ್ಲ ಆ ಟೈಪ್ ಆಫ್ ಗ್ಲಾಸ್ ಸ್ಪ್ರೆಡ್ ಮೇಲೆ ಅದ್ರ ಮೇಲೆ ಒಂದು ಡ್ರಾಪ್ ಆಫ್ ಕರ್ಡ್ ಆದಮೇಲೆ ಹಾಕಂತ eventually heat and prepared a thin smear. Okay, dry the smear by heating fixation by passing the slide 5 to 6 times on the spirit lamp. So ಅದನ್ನ ಡ್ರಾಪ್ ಆಫ್ ಮಿಲ್ಕ್ನ ಏನು ಮಾಡ್ಬೇಕಂತ ಒಂದು ಸ್ಪಿರಿಟ್ ಲ್ಯಾಂಪ್ ತೊಗೊಂಡು ಅದನ್ನು 5 to 6 minutes ಬಿಟ್ಟು ಅದನ್ನು ಡ್ರೈ ಆಗಕ್ಕೆ ಅಂದರೆ ಒಣಗಾಕ ಅದನ್ನು ಬಿಸಿ ಮಾಡ್ಬೇಕಂತ. ಬಿಸಿ ಮಾಡಿದರೆ ಅದು ಏನಾಗ್ತೈತಿ, ಡ್ರೈ ಆಗ್ತೈತಿ. ಸೊ ಆ್ಯಡಿಂಗ್ methylene blue stain on the curd smear and leave it for 1 minute. ಸೊ ಅದ್ರ ಮೇಲೆ ಒಂದು ಬ್ಲೂ ಮೆಥಿಲಿನ್ ಹಾಕ್ಬೇಕಂತ, ಬ್ಲೂ ಕಲರ್ ಮೆಥಿಲಿನ್ ಅದ್ರ ಮೇಲೆ ಹಾಕ್ಬೇಕಂತ. ರಿಮೂವ್ ಎಕ್ಸೆಸ್ ಆಫ್ ಸ್ಟೇನ್ಸ್ ಬೈ ವಾಷಿಂಗ್ ದ ಸ್ಲೈಡ್ ವಿತ್ ವಾಟರ್. ಸೊ ಈಗ ಹಾಕಿ 1 minute ಅದನ್ನು ಬಿಡಬೇಕಂತ. ರಿಮೂವ್ ಎಕ್ಸೆಸ್ ಆಫ್ ಸ್ಟೇನ್ಸ್ ಬೈ ವಾಷಿಂಗ್ ದ ಸ್ಲೈಡ್ ವಿತ್ ದಿ ವಾಟರ್ಲೇ ವಾಶ್ ಮಾಡ್ಬೇಕಂತ. ಅಬ್ಸರ್ವ್ ದ ಸ್ಲೈಡ್ ಅಂಡರ್ ದ ಕಂಪೌಂಡ್ ಮೈಕ್ರೋಸ್ಕೋಪ್. ವಾಶ್ ಮಾಡಿ ಕಂಪೌಂಡ್ ಒಂದು ಮೈಕ್ರೋಸ್ಕೋಪ್ ಮೇಲೆ ಆ ಒಂದು ಗ್ಲಾಸ್ ಪೀಸ್ನ ನೋಡಬೇಕಂತ. ಅಂಡರ್ ದ ಲೋ ಟೆಂಪ್ರೇಚರ್, ಲೋ ಪವರ್ 10x and high power 1040x and oil emission 100x objectives. Thin objects you see are the bacteria which convert milk into curd. ಸೊ ಅಲ್ಲಿ ಅಷ್ಟೆಲ್ಲ ವಾಶ್ ಮಾಡಿ ಅದನ್ನು ಟೆಂಪ್ರೇಚರ್ ಹೈ ಇಟ್ಟು ಲೋ ಇಟ್ಟು ನೋಡಿದ್ರೆ ನಿಮ್ಗೆ ಅಲ್ಲಿ ಮೈಕ್ರೋಬ್ಸ್ ಕಾಣಿಸ್ತಾವೆ. ನಿಮ್ಗೆ ಇರುವಂಥ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್, ಸಣ್ಣ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಇರ್ತಾವೆ ಅಂತೇಳಿ ನಿಮ್ಗೆ ಕಾಣಿಸ್ತೈತೆ. ಎದ್ರಾಗ ಕಾಣಿಸ್ತೈತಿ, ನೇಕಡೆ ಕಾಣಂಗಿಲ್ಲ, ಅದನ್ನ ಮೈಕ್ರೋಸ್ಕೋಪ್ನಾಗ ಕಾಣಿಸ್ತೈತಿ. ದೇ ಆರ್ ರಾಡ್ ಶೇಪ್. ಹೆಂಗೆ ಅದಾವ ನೋಡು, ತೋರ್ಸಕತ್ತಾರ ನೋಡಿ ಇಲ್ಲಿ ಬಾಜು ಪಿಕ್ಚರ್ನಾಗ. ರಾಡ್ ಶೇಪ್, ಥಿನ್ ರಾಡ್ ಶೇಪ್ ಅದಾವ ಅಂತ. ರಾಡ್ ಶೇಪ್ ಬ್ಯಾಕ್ಟೀರಿಯಾಸ್ ಕಾಲ್ಡ್ ದ ಬ್ಯಾಸಿಲಿ. ಈ ರಾಡ್ ಶೇಪ್ಡ್ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಏನಂತ ಕರೀತಾರೆ? ಬ್ಯಾಸಿಲಿ. ಏನಂತ ಕರೀತಾರೆ? ಬ್ಯಾಸಿಲಿ. singular bacillus. The bacteria that you have observed in the curd samples are the bacilli lactobacillic. So curd ಒಳಗೆ ಇರುವಂತ ಬ್ಯಾಕ್ಟೀರಿಯಾ ಹೆಸರು ಏನೈತಿ? lactobacilli. So here are they bacteria of different shapes? Yes, they exist in different shapes. Bacillus rod shaped, coccus spear shaped, spiral shaped, and vibral curve shaped. And different types of shapes different types of shapes of bacteria ನಮಗೆ ಕಾಣಿಸ್ತಾ ಇದೆ. By observing the figure 7.6 we can know that the colic bacteria may be single, in pair, in cluster, and in chain. Comma shaped bacteria are called vibral. Spiral coiled bacteria are called spirally. So like that, if you can see in the figure they have given here, these type of bacteria are there like this. Okay, they have given this is rod shape, this is curved shaped bacteria. Like that, you can see here there are different shapes of bacteria in our. You can see there's different shapes of bacteria. Yes, you can see here the structure of a typical bacterium. You can see here what parts are there here. This is a pili, here is the mesosomes, yes, cytoplasm, cell wall, cell membrane, ribosome, the phaglium, and the DNA nucleoid. These all things you can see in a bacteria. Okay. Yes, next we are studying about virus. Next part is, now first one we study about the bacteria. Now let us study about the virus. You know virus, you have worldwide you have heard virus in this 2020 year you have heard so many times. Till we are seeing about the virus in TV. What is the name of that virus, children? Yes, it is coronavirus. Yes, let us talk about what is a virus. Okay, now, virus are non-cellular entities that consist of nuclei and acidic core. So what is virus here? This virus, it is not good for our health. Like bacteria are helpful to our body, this virus they are not helpful to our body. They are reducing our immunity power, and they are damaging our body, and they are giving some disease, external disease to our body. So they are not having any cells. It is a non-cellular. Okay, it is a non-cellular entity which is considering a nucleic acid core. What it is consisting? It is consisting nucleic acid core. So DNA or RNA surrounded by a protein coat called capsid. Nucleic acid and capsids together form a nucleoside. So the nucleoside may be naked or surrounded by loose membrane called envelope. So how is the virus he has described here? Virus contains only one type of nucleic acid, either DNA or RNA. Here is saying virus, it is not having cells, it is having only nucleus. Virus is having only nucleic acid. What it is having? It is having nucleic acid. That nucleic acid, it might be DNA or RNA. Anything of this both. DNA or RNA. Virus are infectious agents. What I told you just now, virus, they are not causing anything helpful to us, but they are causing harmfulness to us. So it is an infectious agent. They are much smaller than the bacteria. Bacteria they are more smaller. The approximately size range from 20 to 30 mm. So nm. So 20 to 30 nm. They can be seen only through the electronic microscope. Okay. So examples of virus. Here he has given you about the examples of virus. Virus are incapable of independent growth in the artificial media. So virus are incapable of independent growth. They are incapable. They cannot grow by themselves. They need the help of a body. Then they will grow. Tantane adu develop agangilla. ಅದಕ್ಕೆ ಏನು ಬೇಕು? ಒಂದು ಹ್ಯೂಮನ್ ಬಾಡಿ, ಆನಿಮಲ್ ಬಾಡಿ ಏನಾರು ಬೇಕು, ಆವಾಗ ಅದು ಮಲ್ಟಿಪ್ಲೈ ಮಾಡ್ತದೆ. ಸೊ ಅದಕ್ಕೊಂದು ಗ್ರೋತ್ ಇನ್ ಎ ಆರ್ಟಿಫಿಷಿಯಲ್ ಮೀಡಿಯಾ. ದೇ ಕ್ಯಾನ್ ಗ್ರೋ ಓನ್ಲಿ ಇನ್ ಎ ಆ್ಯನಿಮಲ್ ಆರ್ ಎ ಪ್ಲಾಂಟ್ ಸೆಲ್. ಎಲ್ಲಿ ಬೆಳಿತಾವಂತ? ಇನ್ ಎ ಆ್ಯನಿಮಲ್ ಆರ್ ಎ ಪ್ಲಾಂಟ್ ಸೆಲ್ ಒಳಗೆ ಬೆಳಿತಾವೆ. ಆರ್ ಇನ್ ಎ ಮೈಕ್ರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ಸ್, ಯಾವುದೋ ಒಂದು ಮೈಕ್ರೋ ಆರ್ಗ್ಯಾನಿಸಮ್ನಾಗ ಆ ವೈರಸ್ ಬೆಳಿತೈತಿ. ಹೆನ್ಸ್ ದೇ ಆರ್ ರೆಫರ್ಡ್ ಆಸ್ obligatory intracellular parasites, because they cannot develop their body themselves. It is taking some other external media to artificial media to develop, so it is called as the obligate intracellular parasites. Okay, parasites means who are depending on others for their food or for their livelihood. So that type of organisms are called parasites. So they lack microbiologic machineries of their own to generate energy. What they are doing? They are giving out they are giving some metabolic, they are giving something out to spoil the body. So they lack metabolic machinery of their own or generate energy or synthesize proteins. They depend on the host cell to carry out these vital function. ಸ್ವಲ್ಪ ಹೌ ಮೀನ್ಸ್ ಸ್ವಲ್ಪ ಮಂದಿ ಮೇಘಗಳಿರ್ತಾರೆ ನೋಡಿರಲಿಲ್ಲ, ಮೇಘಗಳಿದ್ದವ್ರು ಏನು ಮಾಡ್ತಾರೆ? ತಮ್ಮ ಕೆಲಸ ತಮ್ಮ ಫ್ರೆಂಡ್ಸಿಗೆ ಒಪ್ಪಿಸಿ ಅವ್ರು ಆರಾಮ್ ಕುಂದಿರ್ತಾರೆ. ಬಟ್ ಕೆಲಸ ಮಾಡ್ತಾರೆ, ನಾನೇ ಮಾಡಿದಂತೆ ಹೇಳ್ಕೊತಾರೆ. ಓಕೆ, ಅದೇ ಟೈಪ್ ಈ ಒಂದು ವೈರಸ್ ಏನು ಮಾಡ್ತೈತಿ? ಬೇರೆ ಏಜೆಂಟ್ದು ಕೆಲಸ, ಅದ್ರದ್ದು ಕೆಲಸ ಎಲ್ಲ ಬೇರೆ ಏಜೆಂಟಿಗೆ ಒಪ್ಪಿಸಿ ಅದು ಆರಾಮಾಗಿ ಇರ್ತೈತಿ. ಸೊ ಇಟ್ ಡಿಪೆಂಡ್ ಆನ್ ದಿ ಹೋಸ್ಟ್ ಸೆಲ್. ಎಲ್ಲ ಬೇರೆ ಸೆಲ್ಸ್ನಾಗ ಅದು ವೈರಸ್ ಎಂಟರ್ ಆಗಿ ಏನು ಮಾಡ್ತೈತಿ? ಅದು ಸುಮ್ ಕುಂತ್ಬಿಡ್ತೈತಿ. ಸೊ ಇಟ್ ಡಿಪೆಂಡ್ಸ್ ಆನ್ ದ ಹೋಸ್ಟ್ ಸೆಲ್ ಟು ಕ್ಯಾರಿ ಔಟ್ ದ ವೈಟಲ್ ಫಂಕ್ಷನ್ಸ್. ದೇರ್ ಈಸ್ ಎ ಗ್ರೂಪ್ ಆಫ್ ವೈರಸ್ ವಿಚ್ ಇನ್ಫೆಕ್ಟ್ ಬ್ಯಾಕ್ಟೀರಿಯಾ. ಸೊ ಗ್ರೂಪ್ ಆಫ್ ವೈರಸ್ ಹೋಗಿ ಏನು ಮಾಡ್ತಾವೆ? ಬ್ಯಾಕ್ಟೀರಿಯಾಗ ಇನ್ಫೆಕ್ಟ್ ಮಾಡ್ತಾವೆ. ದೇ ಆರ್ ಕಾಲ್ಡ್ ದಿ bacterial orphage. They are called as the bacterial orphage. These viruses infect the bacteria and multiply the inside bacterial body and cause the lysis of the bacteria. So here DNA virus or the RNA virus means what he has given you here. It is the hepatitis B, pox virus. Pox and the smallpox antaralla. Smallpox antarilla now. Measles, smallpox antaralla. So this virus, the ವ್ಯಾರಿಸುಲರ್ ಹೋಸ್ಟರ್ ವೈರಸ್ ಎಕ್ಸೆಟ್ರಾ. ಈಗ ಹೊಸದಾಗಿ ನಾವು ಅದನ್ನು ಕರೋನಾ ವೈರಸ್ನಾಗೂ ಅದನ್ನು ಬರ್ಕೋಬೋದು. ನೆಕ್ಸ್ಟ್ RNA ವೈರಸ್, HIV, ಇನ್ಫ್ಲೂಯೆನ್ಸಾ ವೈರಸ್, ಪೋಲಿಯೋ ವೈರಸ್, ಮಮ್ಸ್ ವೈರಸ್ ಎಕ್ಸೆಟ್ರಾ. ಇವೆರಡಕ್ಕೂ DNA ಅಂದ್ರೇನು, RNA ಅಂದರೆ ನಿಮ್ಗೆ ಅಲ್ಲಿ ಡಿಫ್ರೆನ್ಸಿಯೇಷನ್ ಗೊತ್ತಾಗ್ತದೆ. Now let us go to the next part. Let us see the structure of the bacterial orphage. Now here he has given the picture of a bacterial orphage. You can see here that how is the bacteria. It is having a head, it is having a neck, it is having a collar, it is having a sheet, it is having a tail, it is having a fiber, it is having a base plate, and this is the DNA cell. Okay, you can see it here in the figure. Okay, so look at the figure 7.9. The bacterial orphage has a head region containing a genetic material. A head part naga genetic material idandre main part ittadi of a DNA. Collar region contains the proteins. Collar region ಒಳಗ protein sheet ಇರ್ತೈತಿ. It has a tail which is hollow tube. It is having a tail which is having a hollow tube, which the nucleic acid pass during the infection. At the end of the tail there is a base plate. ತಳಗ ಏನೈತಿ? ಒಂದು base plate ಐತಿ, and one or more tail fibers attached to it. So the base plate and the tail fibers are involved in the binding of phage to the bacterial cell. Okay, so you can see how it is working here. Next comes the protozoa. Next comes the another type of bacteria, that is the microorganism, that is the protozoa. Protozoa is a unicellular. It is a unicellular means it is a unicellular body. So protozoa are located in the moist habitat. ಎಲ್ಲಿರ್ತಾವಂತ ಈ ಹಸಿ ಏನಿರ್ತಾವಲ್ಲ, ತಂಪಂದು ರೀಸನ್ ಎಲ್ಲಿ ಇರ್ತೈತಿ ಅಲ್ಲಿ ಆ ಆ್ಯನಿಮಲ್ಸ್ ಇರ್ತಾವ, ಅಂಥ ಒಂದು ರೀಸನ್ಸ್ನಾಗ ಈ ಒಂದು ಬ್ಯಾಕ್ಟೀರಿಯಾ ಇರ್ತೈತಿ, ಆರ್ಗ್ಯಾನಿಸಮ್ ಇರ್ತೈತಿ. ಸೊ ಫ್ರೀ ಲಿವಿಂಗ್ ಸ್ಪೀಸೀಸ್ ಇನ್ಹ್ಯಾಬಿಟೆಡ್ ಫ್ರೆಶ್ ವಾಟರ್ ಒಳಗೆ, ಮರೈನ್ ಎನ್ವಿರಾನ್ಮೆಂಟ್ನಾಗ, ಟೆರೆಸ್ಟ್ರಿಯಲ್ ಸ್ಪೀಸೀಸ್ ಒಳಗೆ ಈ ಒಂದು ಪ್ರೋಟೋಜೋವಾ ಇರ್ತೈತಿ. Decaying organism matter. Some species are parasites of plants and animals. So what is a protozoa? Protozoa species are aerobic. Protozoa species ಹೆಂಗಿರ್ತಾವ? ನೀರೊಳಗೆ ಜಾಸ್ತಿ ಇರ್ತವೆ. But some anaerobic species have been found in the human intestine. ಸ್ವಲ್ಪ ಅನ್ಅರೋಬಿಕ್ ಸ್ಪೀಸೀಸ್ ಏನಿರ್ತಾವಲ್ಲ ಅವು ಹ್ಯೂಮನ್ ಬಾಡಿ ಒಳಗೆ ನಿಮ್ಗೆ ಕಾಣಿಸ್ಕೊಳ್ತಾವೆ. they have a cell membrane. ಅದರೊಳಗೆ ಈ protozoa ಏನಿರ್ತದೆ, it has a cell membrane. Many protozoa species move independently by one of the three types of locomotory organisms. So that is the Fagli, Chilii and the Pseudopia. So these are the parts of the of the protozoa. Next, protozoan reproduction by asexual method. So here the reproduction of this protozoa, it is not it is called as an asexual method of reproduction. Sexual reproduction has been observed in several species. Okay, in protozoa there is an asexual method of reproduction. See, the next part I will take in the next class. Till then you read the lesson, children. Okay, till then you read the lesson and try to answer the questions given behind the lesson, and you try to do this drawing and label the parts. You know, label the parts and know about the figure here. Okay, thank you, children.